Assalamu alaikum. I'm Dr. Sharmin Rahman, Department of Anatomy, Adherens Medical College. Today I'm going to discuss about junctional complex and specialization of cell surface. What is junctional complex or intercellular junctions? Several membrane-associated structures contribute to adhesion and communication between cells. They are present in most tissues but are particularly numerous and prominent in epithelial tissue. The lateral membranes of epithelial cells exhibit several specialized intercellular junctions. Various junctions serve to function as cells to prevent the flow of materials between the cells, such as in occluding junctions, sites of adhesion in adhesive or anchoring junctions, or it may act as a channels for communication between adjacent cells, such as in gap junctions. So junctions may be tight junction or junula occludens, junula adherens, then desmosome or macula adherens, gap junction, and hemidesmosome. Tight junction. These junctions are seen in the most typical surface of the cell membrane and completely encircling the cell. Cells are fused without any gap. This is the figure of tight junction, where you can see there is no gap between the two cell membrane. It may be seen in the urinary bladder. Now, junula adherens. Junula adherens situated just basal to the junula occludens or tight junction. It provides adhesion of one cell to its neighbor. Adhesion is mediated by transmembrane glycoprotein of each cell, that is the adherens. Inside the cell, adherens bind the protein catenin, which is linked by actin binding proteins to actin filaments, all of which produce electron-dense plaques of material on the cytoplasmic surfaces of adherent junctions. The numerous actin filaments form part of terminal web. This type of junction we can see in the cardiac muscle, that is known as fascia adherens in the cardiac muscle. Now, desmosome or macula adherens. Cell membrane of each cell is thickened due to presence of a attachment plaque. Groups of intermediate cytokeratin filaments are inserted into the attachment plaque. In this figure, we can see this blue color. This is an attachment plaque, and these are intermediate filaments which are attached with this attachment plaque. This type of junction we can see in the epidermis of skin. Now, gap junction. Individual unit of gap junction is called a connect zone. Each connect zone is formed by 6 gap junction proteins called connexins, which join together leaving a hydrophilic pore about 1.5 nanometer in diameter in the center. In this figure, we can see these are individual unit that is known as connect zone. And we can see each connect zone is formed by 6 gap junction proteins that are known as connexins. And when these connexins are joined together, in the center, there is a gap which is 1.5 nanometer in diameter to pass the substances from one cell to another cell. This type of gap junction we can see in the intercalated disc of cardiac muscle. Now, hemidesmosome. This is the special type of junction which is present in the basal part of the cell that bind the epithelial cell to the subjacent basal lamina. The 4 junctions that we are discussed about that are present in between cell membrane between 2 neighboring cells, and this hemidesmosome is formed in the epithelial cell with the subjacent basal lamina. It is almost looks like desmosome, but here attachment plaque is seen only in one side. Whereas in the desmosome, this attachment plaque, which is also attached with these intermediate filaments, are found in both sides, but in hemidesmosome, we found this attachment plaque in only one side. Now, specialization of cell surface. The apical surface of many types of epithelial cells has specialized structures to increase the cell surface area or to move substances or particles bound to the epithelium. These are microvilli, stereocilia, cilia, and flagella. Microvilli are short or long, finger-like, plasma membrane-bound projections of the cytoplasm from the cell surface. These are also known as brush border or striated border. We can see this microvilli in the small intestine, proximal convoluted tubule of kidney. Functions of these microvilli are to increase the surface area. Now the structure of microvilli. Below the microvilli is the terminal web, a horizontal network of actin filaments and associated proteins including myosin. The actin filaments cross-linked to one another by proteins such as fibrin and vilin. And bound to the plasma membrane by proteins such as myosin I. In this figure, we can see This sickle-shaped structure, these are actin filaments, and the actin filaments are linked with each other by this fibrin and villi, and also they are attached with the plasma membrane by myosin I. Now, cilia. These are cylindrical, motile structure on the surface of the epithelium. Each cilium contains two central isolated microtubules surrounded by 9 pairs of microtubules. The two microtubules of peripheral pairs are joined to each other. Cilia are inserted into the basal bodies. In this figure, we can see these are two central microtubules, and these are 9 peripheral microtubules, which are doublet. One is A, another one is B, and A and B share the theta diameters, and with the A, there is an arm known as dynein. And by the radial spokes, these are attached with the central sheath. Cilia can be found in trachea, bronchi, fallopian tube, and its main function is rapid back and forth movement. Now, stereocilia. These are much longer and less motile than microvilli, and they are branched. Like microvilli, stereocilia also increase the cell surface area and facilitating the movement of molecules into and out of the cell. We can see these stereocilia in the epididymis and ductus deferens. Function of these stereocilia are increased surface area and signal generation in air. Now, last one, that is flagella. These are similar in structure to cilia, but much longer. One flagellum per cell. Only one example of flagella is spermatozoa. So this is all for today. Thank you.